Hello, very good morning, friends. Well, today is 16th July, and we are again here to have a talk on our topic that is the DNA biotechnology and its recombinant DNA. Well, in a short, we have gone thoroughly all over these techniques of the genetic engineering and in a what way this genetic engineering techniques helps today's in our for the welfare of mankind, just like preparing the insulins, preparing a good milk from the cows, meats, having a genetically modified plants which are very resistive for the diseases, and very specific characters, though we uh, can be taken, can be seen in the organism, whatever we desire in the specific organism. Well, this all is taken from the recombinant DNA. What is recombinant DNA? In a very simple words, I would like to say DNA is broken down. That is the plasmid vector which is isolated from the bacteria, which is broken down, and the DNA or the genes from the eukaryotic cells. It might be plant as well as the animal. The desired gene is isolated, plucked from that eukaryotic cells, and it is attached or it is binded with this plasmid vector with the help of these some enzymes. And though the some enzymes are necessary for the breakage of this, and after binding out, it again completes the plasmid its whole circle like structure which is seen in the diagrams. And again this bacterium are allowed to be multiply. There are multiple copies and they are introduced in case of the plants, in the leguminous as well as on the non-leguminous plants, in most of the agrobacterium, tumefaciens, and so on. So, friend, a very common question arises: what is the recombinant DNA technology, and what are the techniques which are used in this recombinant DNA technology? Well, we all are aware of the gel electrophoresis. Gel electrophoresis is a what we can say is a experiment or is a technique which can be isolate the DNA from a solutions or a solute of a specific species. Well, techniques used in the RDT. RDT for recombinant DNA technology. What we see, the gel electrophoresis, the technique separates molecule on the basis of their size. Cast slab of gel materials, usually agarose or polyacrylate. The gel is a matrix of a polymers forming submicroscopic pores. Well, I repeat, the what is the technique of the gel electrophoresis? The technique is that the technique separates in the gel electrophoresis molecules on the basis of their size. The molecules are separated according to the structure or according to their size, not the structure, and cast a slab of a gel material. What is the whatever the material is there, the slab of this aqueous material is casted out. They are differentiated out in a same tube, usually agarose or polyacrylamide is used. The gel is a matrix of polymers forming submicroscopic pores. The next one, the size of the pores can be controlled by varying the chemical composition of the gel. So this is how can one control the size of the pores by the different types, or by the varying out the chemical compositions by using the different types of chemicals in this technique. The gel is set up for electrophoresis. This is the electrophoresis in a tank holding pH buffer. Electrodes apply an electric field. So you can look out at this diagram which is shown over here. Look out at this negative electrode, the positive electrode. Well, the electric field and the direction of a migration is shown on this arrow. Here you see the buffer one, side view of this electrophoresis. Sample is loaded into this well, which is shown in the red way, like a tables like, and this one is the plastic gel box from our right hand sides. So friends, the molecule of separate DNA RNA carry a net negative charges, so they may move along the electric field upwards the positive cathode. What it separates proteins, a detergent would be included which coats the protein with a negative charge. Whereas the larger molecules are held up as they are to pass through the pores of the gel. So they are held up because they don't have the capability to pass out, while the smaller molecules are impeded less and move faster. As the bigger molecule is held up, it moves little bit slower than the smaller molecules. Smaller molecule is active and moves very faster. The result in separation of size with the larger molecules nearer the well and smaller molecules farther away. This one is the well which is shown over here. The smaller molecules will get attached, will get observed here, whereas the bigger will lie somewhere here. So this technique separates DNA molecules on the basis of size, where we spoke earlier also. Volume in solution, which is not necessarily molecular weight. For example, two DNA molecules of the same molecular weight will run differently if one is supercoiled, because the supercoils constrain the shape to be smaller. Two RNA molecules of the same molecular weight will run differently if one has much intramolecular base pairing, make it smaller. So these are two different things in this molecular weight regarding these DNA molecules and the RNA molecules. Aside from the above exceptions, the distance migrated is roughly proportional to the log of the inverse of the molecular weight. Gels are normally depicted as running vertically with the wells of the top and the directions of a migration downwards. So this leaves the large molecules at the top and the smaller molecules at the bottom. Molecular weights are measured with different units for DNA, RNA and proteins. So, well, what we see in this gel electrophoresis, in this technique of the isolations of these DNA molecules, RNA molecules and proteins, we see that the large molecules at the top and the smaller at the bottom. Large will go at the top and smaller will go at the bottom. Molecular weights are measured with different units. Now we'll see DNA. Molecular weight is measured in base pair, or we can also call it as the in short form BP, and commonly in kilo base pairs. So 1000 base pair or kilo base pair. This is regarding about the DNA. We measure the molecular weight in the base pair. Now RNA. Molecular weight is measured in nucleotides and commonly in kilonucleotides, that is 1000 NT or KNT. Sometimes bases or B and Kb are also used. Proteins are used in the deltans, that is the molecular weight is deltans, grams per mole or deltans, and commonly in kilodaltons, 1000 daltons. So these are the units which are used to measure the DNA, RNA and the proteins. Below is a gel strain with a dye. After that is a colored molecule which binds a specific class of a macromolecules in a sequence independent manner. Probably or probes bind in a sequence independent manner. Different strains are also used for different classes of a macromolecules. DNA and RNA are generally strained with ethidium bromide, an intercalcating agent. The DNA ETBR, that is the bromide complex fluoresces under ultraviolet light. Protein is stained with a comacy blue or a silver stain. What is the restriction endonuclease? Endonuclease is an enzyme which helps in cutting a DNA. So, an endonuclease is an enzyme that cuts duplex DNA in the middle, not at an end for exonuclease. Different species of a bacteria have evolved different restriction endonucleases, each to cut a foreign DNA and gets into their cells by mistakes. To be cut, the DNA has to lack their own protein of a protective methylation. There are well over 100 restriction enzymes, each cutting in a very precise way, a specific manner, base sequence of a DNA molecules. So a restriction endonuclease cuts the DNA only at a specific site. Look out at this. This is a notable point. A restriction endonuclease cuts the DNA only at a specific site, usually containing 4 to 6 base pairs. The enzyme has to cut the DNA backbone twice, recognizing the same type of site. Therefore, the site reads the same way, backwards as forwards, and known as a palindrome. Look out at this. Well, we repeat this restriction endonuclease because uh, in this our techniques, in this biotechnological techniques, it plays a very important role in the cutting of the DNA or in the cutting of the plasmids at a specific site. Look out at this. A restriction endonuclease. A restriction endonuclease cuts the DNA only at a specific site where it is required at a targeted sites containing 4 to 6 base pair. The enzyme has to cut the DNA backbone twice. Okay, this enzyme will cut the DNA backbone twice, recognizing the same type of site on the same specific site. Therefore, the site reads the same way backwards as forwards. And such a situation, such a condition, such a phenomenon is to be known as a palindrome. Well, friends, this was about the restriction endonuclease or endonucleases. Well, an endonuclease is an enzyme that cuts duplex DNA, a very common phenomenon in the middle, not at an end. See what it does. It does this endonuclease. What is the work of this endonucleases enzyme? Restriction endonucleases enzyme. It will cut the duplex DNA and at the very specific site, that is the middle site of the DNA. Different species of bacteria are evolved. Different restriction endonucleases, each to cut foreign DNA. Whereas the different varieties, species of bacteria are evolved with these restriction endonucleases to cut out the foreign DNA to get into their cells by mistakes or by To be cut, the DNA has to lack their own pattern of a protective methylation. There are well over 100 restriction enzymes for cutting, each cutting in a very precise way, a specific basic sequence of the DNA molecule. So there are many enzymes and they have their very specific role. Here we see the restriction endonuclease role. It cuts the DNA, duplex DNA at a very specific and on the middle of the size. Look out at this diagram. So this is the restriction enzyme taking action on the Escherichia coli. The bacteria is Escherichia coli. So look out at this enzyme cuts both the DNA strands at the same site. See, this it is cutting the foreign DNA as well as the parent DNA sites on the specific on the same sites. So well we see this cutting of the DNA with this endonuclease. This one is the foreign DNA. Well, we can see the bases, that is guanine, adenine, thymine, cytosine. These are the bases of the nucleotides of this DNA, and this one is the foreign DNA, or we can also call it as this DNA as a target or the desired genes or the desired DNA which we are. So DNA fragments join at sticky ends. So what we see here, these are the two fragments, 1 and 2. These are the two fragments, and these two fragments are to be attached, are to be combined, or they should be what we can say assembled in a same pattern. Half one of this foreign one and half one of this pure one. So this is the sticky end of this enzymes which are shown of this DNA, shown with the, this one is the sticky end of this foreign DNA. So sticky end from the lower side of this DNA and sticky end from the upper side of this DNA can be seen over here, and now they will be going to attach in a proportion way that they would bind the sequences to with each other. So look out at this. So how this recombinant DNA is prepared. So now this is the recombinant DNA. This was the part of the foreign DNA, and this was the part of the what we can say the ancestral DNA. So now it completes the recombinant DNA with this. So what we see here, the sticky ends from two different DNA molecules can hybridize together. Then the nicks are sealed using ligase. So this is the ligase enzymes. The result is recombinant DNA. When the recombinant vector is inserted into E. coli, that is the Escherichia coli. So what has they done with the help of the ligase? They have sealed the DNA, and now it is the result shows that it completely becomes the recombinant DNA. When this recombinant vector is inserted in the bacterium known as the Escherichia coli, the cell will be able to process the instruction to assemble the amino acids for insulin production. This is the what we can say the very useful. More important, the new instructions are passed along to the next generation of E. coli cells in the process known as gene cloning. This is the gene cloning. So what happens in the recombinant DNA? The recombinant DNA is actually bind with this ligase, and after forming the recombinant DNA, this is inserted or introduced through the bacterium, that is the Escherichia coli, and the cell will be able to process the instruction. Now, after inserting in the bacterium that is known as the Escherichia coli, now the bacterium cell is able or has a capability to process the instruction, to give the information, to have the activity, to assemble, to produce the amino acids for insulin, for the preparation of insulin productions. And moreover than this, more importantly, that the new instructions from this bacterium, from this Escherichia coli bacterium, are passed along to the next generation, to the upcoming generation of E. coli cells. And in such a process, such a phenomenon is called as the gene cloning. So, friends, today we have has learned some words or have a topic discussed about the recombinant DNA technology and the restriction endonucleases. What is the recombinant DNA? What is the role of the restriction endonucleases? Why do we go for the recombinant DNA technology? What type of technique is this? How it is manipulated? What helps to recombine the DNA, that is the ligase, and how this DNA is introduced in the agrobacterium or in the Escherichia coli. Well, we saw the example of the Escherichia coli, and after the recombinant DNA is inserted in this Escherichia coli, how it gets a process, it gives the informations to instruction to assemble, to produce the amino acids for the production of the insulins. And moreoverly important is that these E. coli cells overpassed out the instruction to the next generation, to the upcoming offsprings, and such a process is to be known as the gene cloning. I think so. We shall stop here. If you have any doubts, queries, you may contact on my cell phone or on our WhatsApp. Till then, thank you. Goodbye.